Welcome to my sections. What I'm planning to do with these sections is just to record and go over content material like lectures 1 through 3 and just try to give you guys the main takeaways. This is what you should be focusing on. This is how they could test you. And this is not to say this is going to be exhaustive at all by any means. Like, there's stuff that I may not cover here that was in the lecture slides. Like, go back and visit it. But I just handpicked a couple things that I think you should definitely have solidified before you start doing the little granular details and memorizing those teeny tiny things. But yeah, so my plan is to have office hours on Wednesday. I still have to confirm time and rooms and everything, but my hope is to just have these online recorded kind of conference sections for you guys to look at your own pace, 2 times speed, 1 times, watch it while you're eating or whatever. This is just supposed to be something where I can explain the concept in a different way perhaps, or just be able to emphasize what you should be focusing on. And then in the slide I have a bunch of other stuff like grades, achieve homeworks, extra logistical stuff. So keep in mind your exam is going to be, I think today is let me check, today's the 9th, so we have about 10 days. So not this coming Wednesday, but the following Wednesday you guys have your first exam at 6 p.m. And yeah, just hopefully we can get started on all these conference sections. So my goal through these sections is to 1 to 3. So I'm trying my best to cover everything, and yeah, just hopefully I can give you a brief overview of what I think you should be focusing on, explain things in a different way, and better improve your understanding. So yeah, so first thing we're going to work on is very easy, amino acid basics. So as you know, there's an alpha carbon. If I was showing you here, it's this one that's not highlighted or indicated or whatever. My apologies, the diagram doesn't have it. But this alpha carbon, the reason why we call it's a special carbon is because it's connected to 4 different groups, 4 distinct. There's the hydrogen, and there's the R group, which is variant, and if it's glycine, obviously it's going to be another hydrogen. We'll get into that later. Then we have the amino group, which is our amine, so it's an N, and then the 3 H's are 2, depending if it's protonated, deprotonated. We'll go more into that. There's the carboxyl group as well. And so those are the 4 groups connected to the alpha carbon, and basically the carboxyl is going to be acidic. It's going to be the first thing to lose its proton, whereas the amino group is going to probably be the last one to lose it because it's more basic. It wants to kind of hold on to that electron density before becoming protonated. And so just some general like kind of memorization stuff you have to know is proteins only contain L amino acids, and when you're drawing these, like, be very careful of where the orientations of these groups are, because these are technically Fisher projections. And the reason why I say that is because in Fisher projections, you could write them as lines, not like as dashes or wedges, just lines, and it'll represent this essentially, where on out to the sides they're wedges, meaning that they're coming towards you, and then whereas they're going behind the page, they're going away from you, if that makes sense. And so it's good to keep this in mind, and the best like nomenclature, or the best thing to keep in mind as you're drawing it like this, if it's L or D, is to always put the carboxyl group and then focus on where the amine group is at. So we say L, and the mnemonic is like L is for the amine being on the left of this of the alpha carbon, whereas in D we're expecting it to be amine on the right. But this is just a simple mnemonic on how to determine L or D. And yeah, just know that the other 2 groups, the hydrogen will either flip depending if the amine is there, but then our R group is going to be on the bottom. So this is just something to keep in mind as you're distinguishing between L or D, or if, Professor Holmes or Lisi asks you, oh, just draw an L or a D amino acid. You know exactly how to do that with the nomenclature. So there's 20 amino acids. So you should know your 1-letter codes, your 3-letter codes, and then also the structures as well, like the R groups specifically. So if we were given like lysine, for example, this is just an example. Lysine, we know that the 1-letter code is not L because that's leucine, it's K. And we also know that the 3-letter code is LYS, the first 3 letters, which makes it pretty convenient. But then the structure, we know that it's going to be 4 carbons, and we know that there's an amine group that's going to be charged, meaning that it's a basic residue. It's a basic amino acid. And just to clarify ahead of time, residue is essentially the equivalent of an amino acid. We just say residue in the context of a whole protein, if that makes sense. So when we're looking at a protein that has like maybe thousands of amino acids, when we say we're looking at the 700th residue, we're looking at this glutamate residue. It's at that specific amino acid. And you could just say, oh, this amino acid, but we, for some reason, just say residue as well. So yeah, you want to be familiar with their classifications. Are they basic, non-polar, acidic, all that stuff? And it's neatly summarized in this chart right here. As you can see, we have the non-polar aliphatic, meaning that it's not like in a ring, besides proline. Proline is the only exception. And then we have the polar uncharged R groups, which is this on the 5 on the bottom. And then we have positively charged R groups, and then negatively charged R groups, and non-polar aromatic. So these are different because they're not necessarily linear non-aromatic molecules, that they're not like ring structures. These ones are. And then The forms here on this entire table are presented at pH 7. So some of them may be protonated, some of them may be deprotonated. It's just something to keep in mind because pH does matter in this context. And also know some of the special cases. For example, glycine is achiral. Why is that? Because as you can tell, it has a hydrogen as its R group. And in order for a molecule, going back to orgo, in order for it to be chiral, it has to have 4 distinct groups attached to it. In this case, this H and this H are the same, so it's not 4 distinct groups, it's only 3 distinct, but that means it's not chiral at all. So that's why we say it's achiral. Then isoleucine and threonine have 2 chiral centers. So if we go to threonine, right, again, sticking to that principle of chirality, in order for a molecule to be chiral, it has to have 4 different groups. So we look at the alpha carbon, it's connected to a carboxyl group, it's connected to an amine, it's connected to a hydrogen, and all this mess right here that's completely different from the other 3 groups. So that's already 1 chiral center. But the second one is going to be this carbon right here, where it's a hydroxyl group, it's an H, it's a CH3, and then it's all this mess up here. So that's something to note, is that there's 2 chiral centers. And they're next to each other, they're adjacent. And then also isoleucine as well has 2 chiral centers. And isoleucine's right here. It's in the same context. Like alpha carbon is going to be a chiral center because it has 4 different groups. And this carbon right here, where we have a CH3, that's 1. And then CH2 connected to another methyl, that's the second group. Then we have an H. And then finally the alpha carbon, all that mess, that's completely different. So again, these are the 2 amino acids that have 2 chiral centers. And then histidine is conditionally charged, meaning that usually the pKa of histidine, this H right here that's highlighted in red, is around 7. So we say conditionally because it depends if we're talking about physiological conditions, because if it's physiological, it'll be deprotonated or it'll be charged, either or. Can't remember which one, but that's why we say conditionally, is because the pKa of that R group is around physiological pH, then we have to take that into consideration when we're denoting if it's charged or not. Again, all these tables are designated at pH 7, but histidine, it could vary. Cysteine forms disulfide bridges, and so if I find cysteine right here, it forms it because you have this SH. We can get rid of the H. Essentially, it's called oxidizing the sulfur and connect it to another sulfur, another cysteine. So you can form these disulfide bridges. And it's different from methionine because if we go to methionine, if I can find it here, right here, top, I saw it somewhere, right here. So for methionine, you see that's a CH3 group, right? We can't get rid of this methyl group because have you ever heard of a methyl group just leaving off of a sulfur or an oxygen? No, usually it's stuck to it essentially. So we can't form disulfide bonds with the methionine because of that CH3. This S cannot be deprotonated. It can't have an extra electron pair to make those bonds. So something to keep in mind as you do this is that cysteine is the only one that can form these disulfide bridges, not the methionine. And then proline doesn't have hydrogen bonds as a residue because the proline itself, because it forms that ring, the R group forms that ring with the amine group, it can't form those hydrogen bonds because that's already there. You can't necessarily deprotonate or protonate that amine. You can't distribute the electrons to other H bonds nearby because we know that as an alpha helix spins, it's going to donate accordingly as like a stacking thing. So something to keep in mind. And then here's some tips for memorizing 1-letter abbreviations. I'm not going to go through this entire list. I think some of these are intuitive that you can just read it on your own. Like for example, the glue, E, the GLU, E, and it forms together like a glue. And so that's how you can memorize it. But these are extra things that you can use if it becomes a little bit more difficult. It comes down to rote memorization, and there is a YouTube video called by the Organic Chemistry Tutor, and it just goes on how to memorize all the amino acids. He has a beautiful way to put it in terms of memorizing everything, and then it's very easy to do it. So I took some of those, put them in here, but there's some that I haven't described yet that you could, if you need another mnemonic that just works for you, it's there as well. And so some tips on how to draw amino acids, right? So first thing you want to do is start off with the building blocks, right? You want to pay attention to the structures that build off of one another. So for example, for phenylalanine, right? Phenylalanine. So alanine, we know that it's a CH3 group, right? I'll show you in this previous slide. Alanine up here, where are my bases, is a CH3 group, right? And if we go to phenylalanine, right? We see that it could have been CH3, but it's not, it's CH2. That's why we see the alanine part. And then we see the phenyl because it's a phenyl group instead of that H. So you can see how some of these amino acid names are derived from each other. And then tyrosine, I know it's not necessarily phenylalanine hydroxyl, I know it could have been intuitive like that, but we know that adding an OH to the end of phenylalanine creates a tyrosine. So that's just, again, a lot of these things are derived from each other, and so it makes it a little bit easier to memorize. These are non-polar aliphatic R groups, where you see that they're non-polar, they're technically just carbons rearranged, adding more. For example, there's only one carbon here, there's 3 here, there's 4 but in a different arrangement. This one is methionine with the S inside of it. So these are just non-polar. The R groups do not lose their charge or gain their charge. They simply just stay uncharged, and they're hydrophobic as a result. And then lysine, for this one, it resembles a non-polar amino acid with C atoms, but with a basic amine at the end. So for example, this one resembles sort of like methionine. I don't want to throw you guys off, but essentially, if we count the carbons, 1, 2, 3, 4, 5. 1, 2, 3, 4. It's almost like we get rid of the sulfur, change it for a C, and amine at the end. The organic chemistry tutor breaks the lysine, arginine stuff down really well, so I recommend that. Just go on that video. I can post a link to it in the announcements or where I'm posting the slides for these. And then note similarities and make connections. For example, aspartate and glutamate, right? Aspartate has just one CH2 and then the carboxylate anion. Whereas glutamate, it's 2 carbons and then the carboxylate. So it's pretty easy to memorize. Okay, aspartate is the shorter one, and then glutamate is the longer one. That's one way to kind of, if you memorize one, you technically have memorized the other. And same thing with asparagine and glutamine. They're exactly the same as the aspartate, and glutamine is the exact same as glutamine, is the same as glutamate. It's only that you change that carboxylate anion right here to amine. Or an amide group at the very end. So again, a lot of these are derived from each other. They're very similar, and noting these similarities makes it a lot easier to just memorize it. And then the next thing that we have is serine, threonine, cysteine. These are kind of the special polar, but not necessarily charged right away. Or they remain uncharged in solution. So I mean, serine is it's kind of derived from alanine in a way. The CH2, and then we just add a hydroxyl instead of that last hydrogen. For threonine, we hydroxyl group and a methyl added to it as well. So instead of a CH2, we just get a CH and then an extra group and an extra hydroxyl. And then cysteine is just the alanine, but instead of an OH, we have an SH. And these are the only amino acids out of the 20 that contain that S inside of it, inside of the R group. And then with these ones, it's kind of trickier. I would say that it's row memorization, but for tryptophan, what really helps me a lot to draw it is to draw the benzene first at the bottom and then draw this complex mess right here, because the benzene, we know that it's the aromaticity has the alternating double bonds. We know there's one here, no double bond here, double bond, no double. Anyways, point is that that's the foundation. And you know that it's almost like an alanine, it has instead of CH3, it's CH2 with this extra group. And then you just connect and memorize these 3 little things. It just comes down to row memorization. And then arginine, this is very related to lysine, but I don't want to confuse you guys. If you want, go to that. Again, I keep linking the organic chemistry tutor, but that's generally how I learn these things. But I will put a link to it. It has to do with a 4-3-2-1. It's a pretty cool mnemonic. It just makes sense. And then histidine, it's one of those where you just kind of have to have middle group, this ring structure with the 2 ends, the double bonds, the conditional charge. You just have to have that memorized as it is. Moving on to the ionization of amino acids. So every single amino acid has at least 2 ionizable groups. And in this case, on the bottom left of this slide, we see that the first group that can be ionized or deionized is the carboxylate group, the carboxyl group. And essentially, it's going to lose the hydrogen and become deprotonated. And then the next group is that amine group. Remember, an amino acid, if we go back to the very first slide, I'm not going to, it's always going to contain the H, the R, and then the carboxyl group and the amine group. It always will. That's an amino acid. If it doesn't have all 4 of those, it's not an amino acid. So that's the number one thing. So if that's the case, if that's the logic, then it's a universal statement that every amino acid has at least 2 ionizable groups because of the carboxylate and because of the amine. Now, some of them are charged, some of them are acidic, like glutamate, aspartate. Those have an extra ionization group in the R group. And there's these special cases as well that we have to consider. So this is where We have the carboxyl group. It's protonated at very low pHs, and then the amino group is protonated until very high pHs, and it's usually the one that deprotonates last. But there's pKa tables that you have to take into account. No, you do not have to memorize it. Like it'll be given to you, so this will be all intuitive in a little bit. So some amino acids, as I mentioned before, have ionizable R groups, which kind of alter the Zwitter ionic form, the pi, the isoelectric point. And just to clarify, a Zwitter ion is having both positively and negatively charged groups. So from a low pH, meaning it's very acidic, there's a lot of protons in solution, a lot. So it's going to obviously protonate everything about the amino acid because there's just so much there. The amino acids are cationic from low pH, and then they turn into Zwitter ionic because you're going to deprotonate the carboxyl group. This carboxylic acid, you're going to deprotonate it and form that negative charge, but you still have that positive charge on the amine. So this is what a Zwitter ion means, is that you have both the negative and the positive charge within one molecule. And then as you increase the pH and you add more hydroxyls, you decrease the concentration of the of the H plus atoms or ions. When you reduce those ions, you're going to basically deprotonate more of the protein, right? So when you're going from this middle protein right here to this one, we're removing so much of this H plus and we're adding OHs in its place to increase the pH. And this should make sense based on the pH is equal to negative log of the concentration of the hydronium ions and all that stuff. Like, it should make sense. But as you get rid of this and you lower that concentration, the pH is going to go up. It's just a relationship. I can come to my office hours if you want me to explain it more in depth, but this is just like a very simplified version of it. And isoelectric point, it's a little bit different from Zwitter ionic, where isoelectric is when the pH at which the molecule, the net charge, is 0. And for amino acids that only have two ionizable groups, like alanine, glycine, the nonpolar R groups, those are going to have a pi that's in between, that the pi is the Zwitter ionic form, right? It's this. But for example, if you introduce glutamate, right, it has 3 deionizable groups. And what ends up happening is there's multiple areas where it's Zwitter ionic, if that makes sense. For example, let me put this here, right? No, it's glycine. Here it is. Okay, so I'll just use this example from histidine really quick. I'll go back into the slides. But what I'm trying to say about that Zwitter ion and pi are separate concepts is that, for example, this charge right here, this histidine on the middle, is Zwitter ionic because it has this negative charge and it has this positive. And this one is also Zwitter ionic because it has a negative and positive. But this one isn't because all of the groups are now negatively charged. There's no positive at all. Does that make sense? Like, pi is a little bit different because if you say this is pi, it's not, because the overall charge is +1 because of that R group. Whereas this one, it is pi because now it's isoelectric. It's overall charge of 0, but it's still Zwitter ionic. So I just wanted to make that subtle distinction between Zwitter ion and also what it means for it to be isoelectric at an isoelectric pH. So let me go back to the slide. So again, you do not have to memorize the pKa values. This table will be given to you. It's extremely unfair if the professor's like, nope, you have to memorize this, because even on the MCAT, you're going to get tables like this to help you out with calculating isoelectric point, calculating all that stuff. And just a quick little note, this is not me trying to say this will not be on the exam. This is just me saying I don't recall any exam where we were asked to deprotonate cysteine, serine, threonine, or tyrosine. I don't recall it, but I mean, it could very well be tested. So always something to keep in mind. Like, for example, we see cysteine here, right? On the pKa table, 1.7, 10.8, 8.3. We'll draw this thing right here where I'm about to show you. Draw it like this. Draw all the states that could be protonated, deprotonated based on that pKa table, because it'll tell you which one gets deprotonated and protonated first. When you draw them like this, then calculating the pi is super easy, and you'll be able to find out what's the Zwitter ionic forms, what is the isoelectric point, what is the structure at an isoelectric point. It's going to be very helpful to have that. And then in terms of calculating PIs for amino acids without ionizable R groups, this is for nonpolar, for the aromatic ones that don't get deionized. If that's the case, where the amino acid only has 2 ionizable groups, then the pi is going to be the deprotonation or protonation of the amine group, that pH or pKa, and the pKa of the carboxyl group. And you're going to take the average of these two, and that'll give you the isoelectric point. So it's pretty straightforward. And just kind of a bonus, what amino acid is this? I'll give you 5 seconds, or you can just pause the video now. But the answer is the amino acid is glycine, because we have these 2 H's. And what I'm trying to say is with this is that again, just draw out the amino acid. Like, this is the best surefire way that you don't miss points on this, is draw the amino acid structure fully protonated, deprotonate one at a time until everything is deprotonated, and then from there figure out which one. Okay, figure out the overall charges. This one is all protonated, so it's obviously going to have a positive, right? If it's histidine, if it's in a basic group, you know it's going to have that extra positive charge, our group, so it's going to be +2. But point is that draw this first, then draw it deprotonating once at a time, and then note the pKs and note the overall charges. Because, I mean, for this example right here, glycine, it's very easy, it's straightforward. But when you get our group that is ionizable, it's very helpful to have it all written out. And if you have a polypeptide, for example, right? I have an example of this later in the slides, where you have to calculate the overall charge, the isoelectric point, and you have multiple amino acids that some of them are ionizable, some of them are acidic, some of them are basic residues. You need to be able to draw the whole structure deprotonating at a time. And of course, doing it this way will require more work, but it is much, much more worthwhile than trying to just do it all in your head. So this is my recommendation, and easy. This is what I've been doing for every single exam when I was tested on it, when I took this class. So kind of going over a more complicated example is a basic residue, histidine. So I gave you the pKs here. pK1 is 2, pK2 is 9, pKr of the R group is 6. And so right here, like I said, if I was doing this, I would draw the structure first, all of it protonated. And the first thing is, what is first deprotonated, right? Given the pK tables, given what I gave you here, right? pK1 is 2. And that 2 is going to not correspond to the amine, because we know that the carboxylic acid is going to be deprotonated first because it's an acid. It can, in the definition of an acid, is it can hold on to that negative charge. It can hold on to that electron density much better. It can stabilize it, obviously through resonance. We don't need to go over that again. But this is going to be the first one that deprotonates, right? So in this instance, I would draw the structure again, but instead of having that carboxylic acid protonated, it'd be deprotonated, and it would have that negative charge. This is now a Zwitterion, but the pK1, this transition between the two, is 2. So I would write above the arrow like pK1 equals 2. And then the next thing is I would take the structure, draw it again, but what's the next group? And if we work our way up, which is the next number after 2? It's 6. And this is for our R group, right? So our R group, the histidine, is going to be deprotonated at this point. So now we just get rid of the H, and now this is not necessarily negative charge. It just has no charge. And the overall charge of this, let me do this, is going to be 0, right? Because this lost its positive, this is negative, this is positive. So 1 minus 1 is going to be 0. So this is going to be an overall charge of 0. And the pKr is the transition between these two, and that'll be at 6. So I would write this is equal to 6. And the now isoelectric structure, this is what it's going to be, the isoelectric point. We don't have the value per se calculated, but we take the averages. And I'll go into that. And then pK2, now I draw the final structure. What's the last thing that's deprotonated in this? It's the amine group right here, this N group. So we're going to deprotonate it. It's going to become a 0 charge rather than a +1. So now the overall charge is negative. We have deprotonated every single group. It is done. And we now need to calculate the PI, right? So this is what I have told everyone in my office hours, is that when you have all the structures written out, all the possible charge combinations, right? What you're going to do is you're going to start from the 0 because that's isoelectric. We're going to go to +1, right? We're going to take this transition. So whatever the pK is, for whatever the pKa of this is, this transition from 0 to +1, we're going to take that into account. And then we're also going to take the pKa of 0 to -1. It's just that. We don't take this one into account, the +1 to +2, because that's not isoelectric. That's not how we calculate it. So we'd only take these two right here. And so what that means is the pKr is 6. So 6 plus 9, so that's 6 plus 9 is 15. 15 divided by 2 is going to be 7.5. And so that's our isoelectric point, is 7.5. So in order to get 100% of the 0 overall charge amino acid right here, in order for this to be at a concentration of 100%, the pH has to be 7.5. That's what isoelectric point is. So the answer is right there. But I kind of wanted to go over this graph because they may ask you to draw it. Just as it is, they'll give you the pK, so you don't have to have that memorized, and you just need to do your best to not necessarily copy the exact curve. You have to know the fundamentals, and I'll just go over it really quickly. And that's if we're starting. I always recommend everyone start from a low pH. What does a low pH mean? It means you have a solution full of hydronium ions, H pluses, and because we have a lot of those, it's going to protonate every R group, every group that can be ionized, it's going to protonate it because we have an abundance of H pluses, right? So this is going to be protonated. This is going to be protonated, and this one as well. So when we have a low pH, let's just say 0, right? Everything will be protonated, and this is at 100% at a 100% quantity, right? So concentration. As we work our way to the pKa, right? Right here we have 1.82. We'll just say it's 2, right? For simplicity's sake. When we're at the pKa, what does that mean in context of these structures, right? So we're taking into account this transition from +2 to +1. We're deprotonating. And the only way, and this is where the OH equivalents come in, when we're increasing the pH, we just add more OHs. It's not that we take out the H pluses, we add more hydroxide ions into it. And so this point is the half equivalence. And half equivalence means that we just added half of the OHs to take this from 100% concentration to 50%. And if this is 50%, the reactant, this +2 charge, then it's 50% the +1 because the OH has deprotonated half of the carboxylate groups. Does that make sense? So what I'm trying to say is that as we work our way to the pKa, we're adding OHs until we reach half the equivalence, so that now instead of having 100% this at pH 0, we come up to the pKa and now we have a 50-50 split. And if you just put it into the Henderson-Hasselbalch equation, you do, let's just say the concentration is 1 to 1, right? It's 50-50, it's equal. 1 divided by 1 is 1, right? And if you take the log of that 1, it's going to be 0. So then your equation becomes pH is equal to pKa. So the pH right here, this y-axis, is going to be the same at half the equivalence, because half equivalence OHs means that you have half the concentration of this and half of this. It's equal. And that means that the pH here is going to be your actual pKa. So that's what it means. And then when we move on to the graph from half equivalence to 1 equivalent, that means we've added enough OHs to deprotonate the starting molecule so that this is 0% now. We've deprotonated every single one, and this becomes our 100% concentration. So that's what 1 equivalence means, is at this point, we now have 100% plus 1 charge. And so this looks about like 4. So if we were at a pH 4, we can expect this to be at 100%, right? So working our way now from there, we have 100% concentration here. Now we're forgetting about this starting molecule. So as we work our way to now 1.5 equivalents, what does that mean? We're adding half equivalents. Again, to that point, instead of having 100% of this now, we're adding half of the OHs, so this gets halved. So now this is 50%, and this also becomes 50%. So now at this pH, we now have a 50-50 split, and this pKa is going to be equivalent to the pH at this point, right? So the pKa is going to be 6, which is the pH at which we've added 1.5 equivalences of OH. And so at this pKa, we're at 6, and we're at a 50-50 split. This concentration here, this molecule, this +1, is going to be the same concentration here at the 0, if that makes sense. And then in terms of isoelectric point, this is where the 2 equivalence comes in. So the more we add, the more concentration, the more this concentration increases, the 0 overall charge. So as we go from 6 and this 1.5 equivalence, as we go to 2, we're now adding enough OHs to get rid of this concentration, this +1, to 0. This is going to be 0 now. There's nothing left of this, and it's going to be 100% this 0 charge. This is the isoelectric point. This is what that overall 0 charge is. That's what the PI is. So if we go to 2 equivalents, this is about 7.5. I mean, we've calculated, it doesn't like necessarily look like it at first, but it's 7.5. And so at pH 7.5, we've added, we basically, this is at 0 equivalence, and this is at 0 equivalence, and this is at this is at 0 concentration, this is at 0 concentration, and this is the only thing remaining in solution. So this is our isoelectric point. This is the pH at which our overall 0 charge species, amino acid, is at 100% concentration, if that makes sense. I know I'm using abstract terms. This is just my best trying to explain it. And then when we work our way up to the last pKa from 2 to 2.5, that means we have taken this 100% concentration and have deprotonated only 50%. So now it's a 50-50 split, a 1-to-1. And so when it's here, then we're at a 50-50 from 0 to -1, and that is our final pKa. That is how this works. And then the more we add to this curve, the more the pH rises most. And the reason why this curve, like in terms of the shape, right now we'll talk about the shape. The reason why the shape goes like almost zero, not necessarily zero, but the slope decreases so much, it's because at this pK we have now a buffer system. When we're at 50-50 concentration, if we add a hydroxide, it's not going to just increase the pH substantially like here, right? It's going to be able to resist these changes to pH, and that's because of the buffer system and going from this charge to this charge. If this is a 50-50 split, that ratio doesn't change that much if you add just a little bit to the reactants or if you add a little bit to the products. The ratio doesn't change. Therefore, the pH or the system's buffer is resistant to those pH changes when you add the OHs. Hopefully that makes sense. But this is me trying my best to explain this diagram on how it works and the shape as to like why it's like curvy. Sometimes it's a little bit flat. Sometimes it's really steep. It's because of that. Hopefully I explained that really well. Come to my office hours if I didn't, or ask questions and whatnot. So hopefully like at least you guys got a sense of how this chart works because I know it's like one of the hardest things in this class when I was hardest things in exam 1 that it was just extremely difficult for me to learn at first. And I would say first, before we move on, make sure, please make sure that you understand this entire concept first, the way I just explained it, or at least what I'm trying to, like the fundamentals, before you use these like shortcuts right here. Calculating PIs with more than 2 pK values, because we're giving you some things to save your time, and you just need to know the acidic and why we're picking these 2 pK groups, the R group and the carboxyl group, and then dividing it by 2 to calculate the isoelectric point for acidic amino acids. It's good to know the fundamentals before you start picking up these shortcuts. So it's really good to work through an example with a basic, an example with an acidic, and an example like cysteine, right? And then also like a glycine or alanine. When you work through all 4 of those examples with the titration curves, you're going to understand this much more than a typical person just trying to memorize the equations. So the PI for an acidic acid is aspartate and glutamate, and they start off with an overall charge of +1 at a very low pH, meaning that if we were at 0, it would be positively charged because we have so many hydronium ions. The H+ is in there. And it takes only 1 depronation to get to neutral. So if you were to do the structures, like I said again, start from the fully pronated, which is the first group that goes. It's usually the carboxylic group on the alpha carbon. That's the first one. And then it's the R group, which by that point, the R group is negatively charged, right? So it's not naturally positive, so that's why it takes only 1 depronation to get to neutral. And if just write this out for yourself and you'll see the overall charges, why it only takes 1 depronation rather than 2 for our basic amino acids. And yeah, and then only 1 more depronation to change the charge to -1, and this is the second pKa value. So then from there you can basically stop and calculate the isoelectric point. And then yeah, and again, overall charge becomes neutral at a lower pH, and this makes sense. It's acidic, it's supposed to be able to be negatively charged at low pHs, if that makes sense. So this like conceptually makes sense why the pH and PI are going to be lower than as opposed to a basic where they love the positive charge. They can hold on to the hydronium ions and all that stuff, and their PI is at a higher pH. So you can read the slide more in depth, take these shortcuts as you will, but if you understand the fundamentals with the pK tables, drawing these structures, it's going to become much more intuitive. And so kind of to like recap, right here we see that I gave you some pKs. We have 3.6, 1.88, 9.6. If we were starting at a pH of 0, at a low pH, this overall charge, we're assuming that everything is positively charged, right? Everything is protonated. It will either be just a +1 or it'll be 0 because it's protonated, and technically we'll lose it once it's deprotonated. And so in this instance, we see that this is going to be the first group that leaves, this H, and then it'll be this group actually that will leave next because it's 3. We work our way up from 0 to 14 or 1 to 14, and then so once this is deprotonated, then the overall charge becomes 0, and then when this becomes deprotonated, then the overall charge becomes -1, and then when this becomes deprotonated, the overall charge is -2 because now there's no positive. This is just a 0 charge, negative. So like again, just draw the structures, make it really easy. You don't have to draw the whole thing out. You can just literally draw the groups that matter and then just do the transitions and which one protonates, deprotonates. And then, yeah, to calculate the PI, you need to figure out which charges allow it to go to 0, right? So from +1 to 0, and then from 0 to -1. So that's the thing you need to keep in mind. And then these ones also go through a similar example. I won't go through all of them. These should be pretty intuitive. now by this point, hopefully. And here's a larger example of a tripeptide now. So draw the following structure from glutamate, lysine, histidine. And remember, this is from the N-terminal to the C-terminal. I'll go more in depth in a few slides, but just something to keep in mind. Your glutamate is your N, so that's going to have the amine-free group, and then your histidine is going to have the free carboxyl group. And so from here, what is the isoelectric point of the tripeptide? And so use that pKa table, write the structures out, which ones can be protonated, deprotonated. The R group specifically is what I'm talking about. And then where's the amine group? Where is the carboxyl group? What is the overall charge at when everything is protonated? And then work your way deprotonating one at a time. And so the answer is on the next slide. I won't go into it as much, but this is the answer. And yeah, the PI is going to be around 7.5 if you take the overall charges from +1 to 0 to -1. Something to keep in mind. Okay, so moving away from amino acids, isoelectric points, let's talk about polypeptides. So polypeptides, in the name, it has peptides in it, and peptides are essentially just polymers of amino acids. Poly means that we have multiple of these polymers combined together into one or multiple subunits. You don't need to worry about that, just the basics. Peptide bonds are essentially the things linking amino acids together, right? So this is highlighted here is a peptide bond. And essentially, what it is that you have a carboxyl group here, you have an amine group here, and it has a double bond characteristic because of resonance, right? Because this amine group can hold on to that extra charge. And so it gives it this partial double bond characteristic. And we'll go more into the properties, the physical stuff of it later in the slides. But this is just something to note that this is what a peptide bond is. And so the N-terminus is the place where the ammonium ion, this amine group, is positively charged, and it's free. It's not connected to a peptide bond at all. It's just, it can be protonated naturally. And then the same thing with the carboxyl group. It's on the opposite end of the polypeptide chain, and it can be deprotonated or protonated because it's free. It's not connected to a peptide bond. And again, like as I mentioned, this is a memorization. Naming is from the N-terminus to the C-terminus, right? So I give you a name like Valhis. Where is the N-terminus? It's the valine, because we just assume it. Unless I say explicitly C, val, his, N, C and N denoting whether it's the N-terminus or C-terminus, unless I make it explicit, always assume that it's N to C. And then in synthesis, something to note is we always construct the peptides from C-terminus to the N-terminus. Just something to keep in mind. And then, as I mentioned, peptide bonds are planar and hard to rotate due to partial double bond character, and I'll elucidate this with a diagram in a couple of slides. And then virtually all peptide bonds occur in a trans configuration, meaning that the R groups are pointed in opposite directions from each other. And that just has to do with thermodynamics, has to do with minimizing sterics, and all that stuff, so we'll go into it. So, Lecture 2, Protein Structures. So first of all, I just want to start off with optical activity of amino acids. So certain amino acids, if they are not, if they are chiral, right, they're going to be able to rotate polarized light in a certain direction, right? And what it means is that polarized light means that all light wavelengths are orientated in the same direction right here. It's in this diagram. So this is unpolarized light, and then when it's passed through, let's just say we shine it through like a little cuvette that contains our solution of just maybe an L amino acid, right? Well, the light is going to curve in a certain way because that amino acid, we're assuming it's chiral, and it'll basically, the polarized light, the light that's able to go through is only one directional, unidirectional. It's in the same direction. When a reaction is racemic, though, this is one thing to know, when it's equal concentrations of L amino acid and D amino acid, there's going to be no optical activity because the light's going to be reflected equally. It'll go this way with the D, this with the L, and it'll cancel, and it won't be, there won't be one linear polarized light direction, if that makes sense. It'll be just multiple directions at once. And so, just to kind of go back into the slides, the negative indicates that there's rotation to the left, meaning counterclockwise, like that. And then, while the plus means that there's rotation to the right, meaning clockwise. And just something like big thing is that this L and D do not dictate rotate direction, meaning that some amino acids, if they're in their L form predominantly, they may be negative, but other amino acids that are also in their L state may be positive. And what this means is that we have to experimentally determine by having 100% L or 100% D what the rotation is going to be. Is it going to be counterclockwise or clockwise? So there's never a general rule of like L is negative, D is positive. There's never ever like something like that. You just have to determine it experimentally, and that will be given to you where they'll say it rotates it left. So what direction is the light rotating? It'll be something like that. As I mentioned, racemic mixtures will not rotate light because you have equal concentrations of the L and the D. Therefore, light will not go in one direction; it'll just go in multiple. And then, this light refractory stuff is used to analyze amino acids and protein structures to determine if you have a chiral molecule, if you have a racemic mixture. There's a lot of things you can do with this. A lot of it's outside of the scope of this course, so this is just kind of like what you should be knowing. So going more into the specifics of this double bond characteristic, we have phi and psi angles. And this relates to the alpha carbon here in the middle, right? So rotation is possible with the bonds connecting the amine group and the carboxylic group to the alpha carbon. So the reason why is because with this partial double bond characteristic of the peptide bond, the rotation is restricted because, again, it has a partial double bond, essentially. And we know that double bonds, they don't like to rotate freely. They have the pi bonds kind of locking that sigma bond into place. And so in this case right here, there's no double bond forming. There's no partial double bond characteristic here. So the rotation is possible. And the best way that it has been explained to me is, I'm doing this on the fly because I know that this will be super helpful, but let me just, there we go. Okay. So the best way that someone has explained it to me, so let me get one piece of paper, is that imagine that the corners connecting these two pieces of paper, right? The corners connecting them is the alpha carbon, right? So if I were to hold it like this, for example, hopefully I don't dilute it, but this corner right here, this corner is the alpha carbon. And so what we say by free rotation is that partial double bond characteristic is going to be like this piece of paper. So as you can see, like on this diagram right here, it's almost like a piece of paper, right? And the corners connecting the two pieces of paper is the alpha carbon. And so what we say about phi and psi angles is how the paper rotates like this. And sorry, let me just, one sec, just cut the papers a little bit. That way it's easier to manipulate. Okay, so let's say we're able to rotate, right? We have this plane. We're rotating around this kind of like two diagonals, this around the alpha carbon, right? That's what the phi and psi angles are. It's not the rotation of the paper itself. It's not like this. It's not like that. What I'm trying to say is that the corners connecting those alpha carbons, that's what the phi and psi angles are. That's what the dihedral angle means, is the pieces of paper stay in a plane, right? But they rotate around the sigma bonds that are connected to the alpha carbon. That's it. That's conceptually what it is. Hopefully that helped. Hopefully it didn't confuse y'all more. But yeah, so there's a mnemonic that I like, and I'll put it on the next slide. It's called FANSAC. And basically, just before I even get into the mnemonic, we know that the phi angle is the bond between the rotation between the alpha carbon and the amine group. And then the psi is the one from the carboxylic acid and the alpha carbon. So that's something you have to keep in to know, is that when we talk about phi bonds, it's this one right here. When we talk about psi bonds, it's this one over here. And there's a mnemonic for it. I'll go over it. And there's a special case. 0 degrees leads to steric clash, meaning that if we were to rotate these molecules, the phi and psi angles, to put them at 0, the back ends of the molecules will clash with each other, and it's not possible to have it because of that steric clash. You can't have two places occupy the same place physically. That's impossible. And to be separate, right? And then 180 degrees are the farthest apart two atoms can be, and that's the trans configuration, right? So different secondary angles will lead to different favored angles, and this is important when it comes to like beta turns, when it comes to the alpha helix. There's a lot of things about that you should know, but this is just like the context of it, the fundamentals. And so again, clarifying with that FANSAC, we know that phi sounds like F, and when you pronounce it, A is for the alpha carbon, N is for the nitrogen. So when we think of phi, fan, so alpha carbon nitrogen, and then sac is psi, we say as pronounced as an S, and then A is for the alpha carbon, C is for the carbonyl carbon, right? So right here's another one. This is Ca for alpha carbon. So the phi is this rotation of the amine group and the alpha carbon, and then the psi is a rotation between the carboxyl group and the alpha carbon. So that's a good mnemonic to know. Don't worry about the omega stuff. That one, it was just part of this diagram, but don't worry about it. For the secondary structure, we know that they're stabilized by hydrogen bonds between amino groups and carboxyl groups, as I mentioned. So right here's an example of this hydrogen bonding. I know this is not a carboxyl group, but this is what hydrogen bonding is. It's hydrogen has fun, F-O-N. Hydrogens can do hydrogen bonding with fluorine, oxygen, and nitrogen. That's all it essentially means. And right here's an example of that carboxyl group doing a hydrogen bond with the amine group. And you see it here through multiple. And that's what stabilizes the alpha helix, the secondary structure. And then we have phi and psi angles, which dictate how protein backbones fold on itself while H bonds hold that 3D shape together, right? So the phi and psi angles will rotate. It won't, because like, again, ideally, energetically wise, 180 degrees is the most stable because it's the furthest apart. If these groups have steric, like, or electronic repulsion, they'd want to be at 180 degrees because that's the furthest apart because they're repulsed by each other. And so sometimes it's not at 180 degrees naturally because of these hydrogen bondings kind of forcing it to not be 180 degrees. That's all that this is supposed to say. So the R groups are not involved in this phi and psi angle dictation at all. The H bond donors are hydrogens bonded to NO for this class, and hydrogen acceptors are NO not bound to H. Essentially. And that's mainly about it from this slide. And then moving on to more this alpha helix, some things you do have to memorize. I mean, it's the most common protein structurally. Like, I mean, DNA is one of the most common. I know it's not a peptide per se, but like just that helical structure, it just goes to show that it's one of the most common ones. But the things you do have to memorize, I recommend that you do if you have extra time, is the 3.6 residues per turn, 5.4 angstroms per turn in case you have to give it in angstroms. I know it's very niche, it's not going to be helpful, but things you will need to memorize. For every alpha carbon within an alpha helix, the phi angle is going to be -60, the psi is going to be -45 to -50. You just need to know the range. That's about it. You don't need to know which exact number. The R groups are going to project away from the core. So if we were to look at it from above, right, like that, or below, whichever you want to say, the R groups are not going to be inside this like circle, right, this loop. They're going to be on the outside, projecting outwards. So that way, because just imagine this for a second, that the R groups did go on the inside. Well, they would clash with each other. Imagine you had a lot of aromatic rings, right? Like that just would not work because they would just keep clashing into each other, and that's not the right way. But if they were on the outside, they can be as long as they want. It could be aromatics, it could be a bunch of stuff, and it won't clash with each other. So that's what our groups do, conformationally are arranged. And so H bonds form between different layers of the helix stabilize it, meaning that the reason why we have the R groups projecting outward in the first place is because it's not 180 degrees, but is in the, what's it called? The hydrogen bonds do stabilize the alpha carbon thing the alpha helix, so that the R groups project outwards. And then the dipole moment has to be neutralized or stabilized. And so we know that from the C terminus, it's going to be negatively charged. Why is that? Because the C, remember that C terminus is the free carboxyl ion. And usually at physiological pH, at pH of 7, that group is deprotonated, meaning it's negatively charged. And then in terms of the amine, at physiological pH, at the N terminus, it's still going to be charged. It's still going to be protonated. So that's why you see this dipole generate within from the C terminus being negatively charged to the N terminus being positively charged. And so you'll typically find positively charged amino acids at the C terminus to counteract that negative charge. Does that make sense? Like you're trying to neutralize it. You're trying to get rid of it. Because if it has a dipole, the protein is not going to be stabilized because it has almost this like unresolved energy imbalance, right? If we would have the negative charge and this is just like physics 40 basics. If you have a negative charge and a positive charge, they want to be together because they're opposite charges. They want to attract. So you need those opposite amino acids at those ends to neutralize that dipole. And then here's the alpha helix destabilization. This comes in terms of identity of proteins, not necessarily this negative charge or anything like that. So there's 5 key amino acids that aren't typically found in an alpha. So proline isn't found in the alpha helix because the phi angle is fixed at -65, so not -60. And its rigidity within the R group connecting to that amine group causes it also to be locked in that -65 degrees. But then also another thing is that because that amine group is taken up by the R group, it lacks the ability to form hydrogen bonds because it doesn't have that free one to donate. And this is what I mentioned before in the first lecture 1 with amino acids. Because the proline can't donate those H bonds, and because alpha helices are stabilized by H bonds, there's a mismatch. And if you put prolines into them, it's not going to be a good outcome. It's going to destabilize the alpha helix. And then there's glycine, which is the opposite of proline, where it is too flexible. It's good for making turns because more angles are possible, but there's a high entropic cost of putting it in, meaning that Glycine, because it's so flexible, because it has that H group inside of it, right, as the R group, it wants to like go in random configurations, right? It doesn't matter where it's at, but we know that the alpha helice, we have those H bonds forming and it causes it to stay stuck, right? But the glycine is just like, I want to move, I want to be flexible. Well, that's what entropic cost means, is that the glycine wants to move, but we're keeping it locked in its place. In order to keep it locked, we need energy to keep it locked in that configuration. So hopefully that makes sense of what it means by high entropic cost. And then there's aspartate, where the negative charge could interact with the hydrogen bonds on the main chain and destabilize the helix structure, meaning that carboxylate ion, let me go back. So for only aspartate, right, and not glutamate, the negative charge of the R group can interact and disrupt hydrogen bonding of the alpha helix. Why is that? Well, we know that aspartate, remember that relation that I told you guys, where aspartate is one shorter than the glutamate, or the glutamate is one carbon longer. So we could argue that this isn't in the glutamate because that R group, that negatively charged carboxyl ion, is so far away from that core, right? If we were looking at the core and the R group is out, it's further away, so it has no chance of like interacting with the backbone, the circular area. Whereas the aspartate may just be here, it may just go out this much, like not that far. So it has the possibility of making that connection because it's close enough in proximity. So again, this can be neutralized with an adjacently positive residue, but more often than not, it could be mostly disruptive to these alpha helices because those H bonds go away. And then we have the bulky aromatics, which is a bulky amino acid, the tryptophan and tyrosine. The reason why they destabilize is because they have a pi-to-pi stacking interactions. So tryptophan and tyrosine, they want to be on top and below each other, almost like forming like a stack. And sometimes that the stacking can cause the DNA helix to turn in ways that destabilize it, if that makes sense. The hydrogen bonding wants to keep it in this circular formation or however it is, but then the stacking may cause it to like open up a little bit to compensate and to form that pi stacking. Something to keep in mind as well. And then moving on from alpha helices, we have to look at beta sheets. There's two types. There's parallel beta sheets. There's anti-parallel beta sheets. So for the beta sheet, R groups protrude perpendicular from the sheet, either above or below the sheet. So we have a beta sheet, right? We're looking at it from the side right here. We can have an R group go above. We can have an R group go below. And there's never an R group going from the sides. It's never going in between like this. No, it's just up or down. And what allows the parallel beta sheets to like stack upon each other is that we have the hydrogen bonding. So sometimes like you'll usually see this where it's an alternating pattern of the R groups, right? R group goes down here, the next amino acid it goes up, the next amino acid goes down, the next one up. But also alternating is that either the carboxyl group is going up or the amine group is going down, right? And it alternates, like right here. This is what I'm trying to elucidate, is that we sometimes may have the amine group pointing down and this carboxyl group pointing up, and this is not the R group, this is just part of the backbone, and that's able to interact with each other to form a parallel beta sheet. Anti-parallel is kind of the same thing, where you still have that interaction, it's alternating, because if we look at this alpha carbon, the R group isn't shown, but we know that it's going up here, and this one's going down, this one's going up, this one's going down. It's alternating in a similar way with these hydrogen bondings as well. So something to keep in mind. So electrostatic interactions provide stability with this. Like if you have an R group, for example, pointing down and an R group pointing up, and one's negatively charged, one's positive, then they're going to interact with each other favorably. They tend to have hydrophobic and aromatic groups, which are found on the inside of proteins. So like in this tin barrel, we would see that the aromatic groups would be on the inside, forming this aggregate of like a hydrophobic region, and they would be concentrated here, whereas these outside helical may be charged or they may be non-polar, and so it just makes sense that they kind of like conformationally, in a 3D sense, they would be closer to each other, closer in proximity, because like interacts with like. Then there's anti-parallel beta sheets, which are more common than the parallel, and they're parallel but opposite directions for their polarity. So, and something to note in here, we see parallel beta sheets. We see a C terminus on the left, C terminus on the left, and they're still able to interact with each other. But the direction from C terminus to N terminus is going that way. It's going in the same direction for both. Whereas in this anti-parallel, we see that the C terminus is on the left, so we're going like this, and then the C terminus on the bottom one right here is going to be on the right, even though it's not listed, the N-terminus. So they're going to go in opposite directions. That's what anti-parallel means, is that they're going anti to each other, but they're parallel, meaning that they'll never like intersect like that. It'll always just be never touching, but going in the opposite directions. That's what anti-parallel and parallel means. And then for the anti-parallel, there's no dipole moment at all. As you can tell here, there's going to be negative charge here, positive charge on the right side because of the C to N-terminus. Here, it's canceled out because you have the negative charge here for this top beta sheet, and then you have a positive charge for this bottom beta sheet. And then for parallel, the polarities are in the same direction. Residues are 6. This apart is something to note. The 7 angstroms apart for anti-parallel, 6 angstroms for the parallel, and which one is more or less stable. Moving on to another secondary beta structure, or another secondary structure are beta turns. There's 2 types. There's a type 1 beta turn. There's a type 2 beta turn. The type 1 consists of cis prolines. Proline is at the +2 position, so 1, 2. This is your cis proline, and this is more common than the type 2 right here. But the type 2 is glycine at the +3, so 1, 2, 3. And the glycine, because it's super flexible, allows for us to do beta turns. That's why. And for the proline, if it's cis, then that means that because the proline is cis, right? Because of proline's rigidity, it allows for it to lock in this beta turn per se, like this phi psi angle to be at -65 for the phi, and then whatever the corresponding psi angle is. And I mean, because it's cis, it's going to be coming towards this amine group, if I'm not mistaken. That's what cis and trans means. But yeah, so this causes the beta turn because of that rigidity. Also something to know is that not all beta turns are type 1 or type 2. There may be some special cases, but these are the ones that you should know. And then there's the Ramachandran plot. So this is a way to energetically visualize the backbone dihedral angles to study protein structure, to tell you what secondary structures are present in it. And it compares the phi psi angles in a protein. So the left axis is your psi, the bottom is your phi. And so correlation is used to estimate the percent of alpha helix and beta sheet in a protein. And if phi and psi angles are plotted, they mostly fall in the alpha helix. It can either be the right-handed or left-handed. Then the protein is made up of a lot of alpha helices. And I'll show you an example in a little bit. And so it's rarely used in labs since other experimental techniques are far, far better. But it just provides an estimation. And they may ask you to interpret a Ramachandran plot. But before I get into the more specifics, we'll talk about supersecondary structures, domains, tertiary structures, and quaternary structures. So supersecondary structures are essentially just a segment of a polypeptide. So think of the primary structure as a chain of amino acids, right? Our polypeptide, it could be linear too. We keep it that. And then we know secondaries are the way that the polypeptide, the backbone, interacts with each other to form an alpha helice, to form a beta strand. It'll interact with itself. And a supersecondary structure is just kind of like a special secondary structure where it's an arrangement of 2 or more secondary structures together, meaning that if we put an alpha, for example, a beta hairpin, right? It's 2 beta sheets, but it's called a hairpin because it forms this 3D conformation. It's not necessarily a tertiary structure, because tertiary is slightly different. In this case, we see that there's a 3D shape that takes place because of that secondary to secondary structure interaction. Does that make sense? Whereas a tertiary structure, I'll just jump a little bit ahead, it's a 3D structure of the whole protein, right? So this beta hairpin may be one little part of this ginormous protein. So we would say that ginormous protein, the way it's folded, that's the tertiary. But when we zoom in on these 2 secondary structures and we see that they interact in a special way, that's a super secondary structure. And so they're non-functional independently, and they're also called motifs and folds. And so kind of distinguishing non-functional independently and the supersecondary structures and domains. Domains are independent functional units of proteins linked by less well-defined connecting bridges. For example, I'm just going to use this example right here, this beta barrel. It may potentially have 4 domains inside of it. One on the top left, like if we were looking at it from top down, right? There may be one on the top left, there may be one on the top right, one on the bottom right, and one on the bottom left. They may have different independent functions that if you knock one out, it gets rid of the enzyme efficiency, right? It makes the protein less efficient. So that's what a domain is it has like catalytic activity, or it has a functional component to it that when you get rid of it, it diminishes the protein's function. And we don't know exactly what the defined connecting bridge is per se. And essentially, like, even if we remove, like, let's say the top strand, and now it's like a free floating protein like this, and it's just in this pocket, it can still function still separately. And so it's like, it could be a catalytic domain, it could be a DNA binding domain, it could be something that is just a functionally independent. And then there's tertiary structure. As I mentioned before, it's, if we look at the protein, we zoom out, and then we look at the way it kind of comes together and folds and all that. That 3D shape is the tertiary structure, and it's stabilized by disulfide bridges and hydrophobic interactions. And we'll go more into this in lecture 3 with electrophoretic techniques. This will become extremely clear in a little bit in terms of the difference between tertiary and quaternary structures. Quaternary are essentially multiple tertiaries coming together. So for example, hemoglobin, we have 4 tertiary structures, if I'm not mistaken. I could be wrong about that, but essentially hemoglobin has 4 tertiary structures coming together to form this quaternary mega protein, essentially. And it's an assembly of protein units put together. So, and this picture elucidates it perfectly. Tertiary structure, we just have 1 protein, but let's say we had duplicates of it, right? And then we combine them to form this mega protein. That's what a quaternary structure is. So hopefully I made clear what a supersecondary structure is, a domain, tertiary, quaternary structure. Hopefully that's a little bit clear. Okay, so we'll talk about some atypical protein folding, and this is just stuff you have to memorize. It's very hard for me to visualize it or like try to explain it. I'll see if I can find some videos or whatnot to explain it better, but essentially with these 2 examples, we have myosin and keratin, which are 2 right-handed helices. So they're 2 separate alpha helices that are right-handed, and they, when they're combined, they twist in a left-handed superhelix, if that makes sense. So the superhelix is like the way they combine and the way they twist. But even though independently, each one may twist in the opposite direction. Hopefully that makes sense. And in myosin, we know that it's a muscle fiber. Keratin is a hair fiber. Keratin makes our hair. Myosin is used for actin, cross bridges, and all that stuff for contracting muscles. So a good thing to memorize is how many residues are per turn. And so this one's 3.5. It's a bit shorter than the alpha helix, which if I haven't memorized by this point, 3.6. Don't quote me on that. Go back in the slides. Then there's one of their properties, special properties, is that they're amphiphilic helices, meaning that they have hydrophobic residues inward, kind of interacting with each other and separating themselves from the outer world. And then you have the hydrophilic regions on the hydrophilic residues, the polar groups on the outside, because they'll interact with the extracellular matrix. And so an example of this is leucine zippers, and those are super secondary structures. And the next thing we have is the collagen triple helix, where you have 3 parallel left-handed helices wrapped around one another in a right-handed superhelix. What does this mean? If we were to take apart the superhelix and have those 3 alpha helices separate, they would be left-handed. And when we combine them, they turn in a right-handed configuration, right? So it's very complicated. I would just memorize how the helices are independently and then how they come together and then what is their directionality. And then also another thing to memorize is they have 3.3 residues per turn. The reason why I say memorize this because they may have like a simple just question like list the number of residues per structure, and they'll say collagen triple helix, they'll say myosin keratin, B DNA, A DNA, that kind of stuff. I'm just giving you an example. So every third residue is glycine near the center. Anything else is too bulky, and so the reason why we're able to have a left-handed stable helice is because of that glycine being super flexible and allowing for us to have this atypical turning. Then there's glycine. The reason why the collagen triple helix also has proline inside of it as well, one, because it's stabilized by the turning, but also because the proline can be hydroxylated and this is a way of post-translationally modifying proline. It allows it to stabilize it much more. And then essentially what's special about this protein is it constitutes 25% of all protein content in the body. So that's truly remarkable how prevalent it is, and it means that, it's necessary for life. I don't know, pretty cool. So here's a practice problem of like a Ramachandran plot. So again, going back to the previous couple slides, or just trying to remember on your own, where would a collagen triple helix occur on a Ramachandran plot, and where would the structure's components fall on it? So what I'm trying to say is, if you have this showing right here, where we have the Ramachandran plot, where would you see the collagen triple helix? We know that's a left-handed individual helix, so we would see it over here, and then the collagen triple helix will show up here, And then in terms of protein folding, right? So there are forces driving folding and unfolding that are related to energy, motion, entropy, and just general thermodynamics and just general thermodynamics. So just two experiments that I'll introduce and then give you the takeaways because that's the most important. There's the Anfinsen, oh man, mouthful. Anfinsen experiment where we add urea and mercaptoethanol to denature a protein and break the disulfide bonds. This will become important in the next lecture. Don't worry about it now, but these are two agents that we added. We remove the denaturing agents and the protein will spontaneously fold back to the catalytically active state every time. So the conclusion is that primary amino acid structure alone is sufficient to determine the native state. What does this mean? When we add these two agents, it denatures the protein. It forms a linear structure now. And that's because we get rid of all the disulfide bonds. We disrupt all the hydrogen bonding with urea. So because we disrupt everything, then what we're saying is, okay, what if we remove both these things? What happens to the protein? Does it fold back? Does it not? What happens? And so what we found out through this experiment is that by getting rid of the agents, the protein starts folding back into the way it was and forms that functional unit again. The protein is functional again. So what the conclusion is that primary amino acid sequence is sufficient to determine the native state, meaning that there's some way to calculate exactly how that primary amino acid will fold in on itself to get this 100% of the time. Because that's what they found experimentally. It's like no matter how many times they did it with linearizing that polypeptide and then getting rid of those agents so it comes back together, it always came back together to that functional state, if that makes sense. And there's this Leventhal paradox, where it means not enough time for a protein to find its native state through a random sampling of all theoretical possible conformations. Meaning that if a protein must be guided in some way folding stepwise. Think of it like, for example, we start from a linear protein going up and down, right? And then it bends one way, and then that eliminates possibilities of it bending this way, right? And then all those other possibilities. So it's almost guided in a stepwise fashion to then form the native state. That's all it's saying. And hopefully that made sense. If not, I can explain it a different way in my office hours. But essentially, it's slowly lower in energy until its native state is reached. So the native state is thermodynamically the lowest possible energy, or the most stable energy that the protein can have in all the possible conformations. And sometimes molecular chaperone proteins, protein disulfide isomerases, peptidyl, prolyl isomerases, assist in folding to prevent aggregation of the protein itself, to maintain that functional capacity of the protein. And then next thing we have is denaturing proteins. So we know that the pH, if we add more, if the pH increases, the pH lowers, it alters the ionization states of the side chains, which subsequently may affect H bonding as well. So, and not even just the side chains, also the amine ends, the carboxyl ends, it changes those too. And so what we're going to talk about is specific molecules like detergents, for example, which associate with nonpolar residues to disrupt hydrophobic interactions. What do we know about hydrophobic interactions? They stabilize the tertiary structure. So if we get rid of that, the tertiary structure gets rid of, the natural state will open up, and now we only have secondary structures holding it together. Then there's high temperatures, which increases the vibrational and rotational energy of the molecules, and that causes enough we add enough heat to the system where now it's almost like that glycine situation where the glycine wants to move. It wants to go all over the place because it's flexible. Same instance here with adding more heat. There's more vibrational frequency. So all amino acids want to move around. And so when we increase the high temperature, we see the denaturing of these proteins. And what TM is the temperature where half of the protein is denatured. Or, alternatively, if you want to think about it more macroscopically, is the temperature where half of all the proteins are denatured. Another one as well. This will usually be you'll be able to determine and distinguish which context you're trying to apply it to. And then there's chaotropic agents, which make more favorable interactions with all residues than water does, thereby disrupting hydrogen bonding. So this is another one that you can use, that you should memorize in terms of like what is able to denature proteins. And then we have the unfolded state under all these conditions has a lower free energy than the native state. What that line means is that The reason why all of these can cause denaturing of the proteins is because we add enough energy to the system so that the unfolded now becomes like the new norm, the new lowest delta G, if that makes sense. And so that's all that this is saying, is that when we manipulate the proteins to either increase the temperature, to increase the pH, to add detergents, to add chaotropic agents, it'll manipulate the delta G of the system so that the unfolded is now the lowest possible, because we're adding external energy or external factors to it. And then the rate of protein folding is nonlinear, meaning that we're not just going to get a stepwise like this. It'll either be like really slow because it's trying to find it, and then really fast, close. Like it's nonlinear. So now moving on to lecture 3, lab lectures, lab techniques, and yeah, this is going to be the final push. So hopefully we can get through it. Oh man, I need to stretch. Okay, let's do this. So we're going to move on to the first technique, which is electrophoresis techniques. And electrophoresis, all it means is it's the movement of ions in an electric field, meaning that if we put an ion in a gel, right, and we add an electric field, the protein will have that the ion will move in correspondence to the voltage, potentials, and all that. So that's very abstract, but that's kind of like the preface of what electrophoresis means. So moving on to this, what's it called? We have SDS page. So the following is going to be added to a protein sample. And this is a very simplistic diagram of what SDS page looks like. But we're going to add SDS, which is a strong detergent, and it also adds that negative charge. And remember, detergents, they disrupt hydrophobic interactions, as in the previous 2 slides. And then we're going to add beta-mercaptoethanol. It's a special molecule that basically reduces disulfide bonds. What do we mean by reduce? We break these disulfide bonds. So this is the oxidized state right here. This SS connection is oxidized. But by adding the H's, we separate these disulfide bonds and they're no longer connected. And this is technically the reduced state as well. And then we also heat the reaction to 100 degrees Celsius, which again, we know that increasing the temperature denatures the protein just because of the vibrational and rotational energies. Again, this is all just kind of the fundamentals. If you understand the basics of denaturing proteins, you'll understand why we incorporate this into SDS page. And so kind of in this diagram right here, we have a multi-subunit. Because we added the SDS page, we add this negative charge all around. And because we add the beta-mercaptoethanol, we're going to basically separate protein A and protein B. Now they're 2 separate functional things, and when we put them through this gel, we run them through a gel, and they're going to separate based on size only, not charge. And the smallest proteins will elute faster, will travel faster further down. And so charge isn't a factor because again, SDS, it's negatively charged. So we're gonna all the proteins are going to be considered negative now. And because we added beta-mercaptoethanol, we're disrupting a protein that is joined with 2 subunits, like a quaternary protein, essentially. And so we form now 2 distinct subunits. Now, these are technically tertiary proteins that are not connected together. I mean, although they're unfolded also. But when we run them through the gel, we have 2 distinct bands. Whereas if we have a single subunit protein, even though we add the beta-mercaptoethanol, there's no disulfide bonds that could be reduced. So it's only going to still end up in 1. So there's never ever an instance that we add beta-mercaptoethanol and we break the actual polypeptide chain. That is not what happens. It's only when we have multi-subunit proteins, when they're connected by these disulfide bonds. That's where beta-mercaptoethanol does its thing and gets rid of it, and it breaks apart the subunits. And then also, there's 2 situations that I've seen on exams, I've seen before, where you add SDS plus reducing agent, and now we're separating based on size. And we also have another situation where we have SDS only, which also separates based on size, but there's a subtle distinction. Remember, reducing agent, what I'm referring to here, is the beta-mercaptoethanol. If we didn't add the beta-mercaptoethanol, this would just be negatively charged, right? But it is still linked together. It is not separated because we wouldn't be able to break this SS bond with just by adding negative charge. That's not how it works. So if in a hypothetical, if we only added SDS and no reducing agent, this would just be 1 band. And it wouldn't even be at B. It would be up here where, let's say D, protein D is just adding these 2 together. It would be further up because it's a larger protein. And then with C, even adding or not adding the reducing agent, it doesn't matter because it's singular subunit. There's no disulfide bonds here. And so it'll be very clear if the protein has a disulfide bond, if it's multiple subunits, if it's a single, it'll be very clear to you. But just know what STS is, what beta-mercaptoethanol is, and what the heat does for STS page in general. Like, just know those fundamentals, and then you'll be able to run through it. And then the reason why it separates proteins on smaller proteins, the reason why smaller proteins travel the fastest is because this polyacrylamide gel has pores that trap the biggest ones, and the biggest proteins are going to have the hardest time traveling through because of those pores. The smaller ones are going to go through it very easily. So something to keep in mind as well. That's why the smaller ones dilute first. And then there's another technique that's also electrophoresis, and we have isoelectric focusing. And so this one separates proteins based on their isoelectric points, where the overall charge is 0. So we run it through a gel, but we're separating based on charge and pH only, not on size, because the proteins move through a pH gradient. We make it size independent by giving it as much time as it needs to get to its isoelectric point. So imagine a gel, right? And we put a pH gradient within this gel. And what we're going to do is, at the top, it's at a pH of 10. And remember, high pH means that there's a lot of hydroxyl groups, hydroxyl ions, and those are negatively charged. So this top is going to be negative. Whereas at a low pH, we have a lot of hydronium ions, H pluses, H3O pluses. Those are positively charged, so we have positive over here. And whatever the protein's overall isoelectric point is, it's going to migrate towards that pH accordingly because it wants to be at a charge of 0. Because again, there's this dipole, not necessarily dipole, but there's this electric voltage difference where we have negative on top and plus on the bottom. And if it's 0, the charge of the protein is 0, then it won't move up or down because it's neutral. So that's something to keep in mind. That's why it separates based on charge, not on size. And then moving on, we have chromatography. And in essence, chromatography is simply just separating samples and purifying your protein or protein of interest or molecule of interest based on some chemical or physical property that they may have. And we'll talk about the specific examples. And in our cases in biochemistry, we usually have an aqueous form of the protein pass through a column. And so there's 3 special kinds of chromatography. There's size exclusion, there's ion exchange, there's affinity chromatography. And we'll go through each single one through here in these next few slides. So gel filtration, or also known as size exclusion chromatography, it separates proteins by size, or length, however you want to describe it, but usually it's size. And this is the difference between SDS page and size exclusion chromatography. SDS page, we allow the smallest molecules to go first. But in this instance, for size exclusion chromatography, it's the largest molecules that go down first. That's the difference. Note that distinction. Write it in your notes. Write it in the slides. Like, this is the distinction. So, like, know that this is complete. This is the opposite, at least the elution rates. This is the opposite of SDS page. So the reason why large fragments travel further in less time because they don't get caught in the channels. So let's say we're taking a little particle here, right, a bead, and what's going to end up happening is we could have a small protein, go through this, and you may say, well, why doesn't it just go through it like fast? Well, the reason why is because let's just say, for example, the protein is shaped like this, like a hook, right, and it matches what's on this thing, right? It's going to be stuck, and it won't move through this little crevice, and it has to be forced through it by other smaller proteins to get pushed through, right? So it's going to take a lot longer to go through here, whereas a larger protein, it'll just go around, but it won't have that resistance. It won't need the flow from other liquids above to push it through, right? Hopefully that made sense conceptually, but again, there's beads in this gel. There's these particles that allow the shorter fragments to fit through. They get kind of stuck. They need other short fragments to push them through, and because it's a mixture solution of large and smaller, it's a variety, the smaller ones are going to have a tougher time because they need that extra push. And so hopefully that made sense that the larger ones elute first, then the smallest are usually the ones that are going to elute last. Then there's ion exchange chromatography, and a lot of students get this very confused. I'm going to try my best to make it extremely clear on what the differences are. The basis is very easy. It separates proteins on charge because the beads that you have on your stationary phase in your column is going to be either negatively charged, the beads, the beads are going to be negatively charged or they're going to be positively charged. And so the column is filled with beads resins with either one of these charges. And what makes it difficult is, like, let's say you have a cation exchange or an anion exchange. What is what? That's what makes it difficult. And so cation exchange means that negative proteins elute first. Anion exchange is the opposite. Positive proteins elute first. And so you can memorize it like this, but I have in the next slide kind of like the cheat code as I like to say. So there's cation exchange versus anion exchange, and I'll clarify exactly how to distinguish between the two. So in the stationary phase, let's talk about anion exchange chromatography, right? So what does it mean? Anion exchange chromatography. This is what I wrote, and I'll start with this actually. If you ever get confused, I put this in quotes, this is me. Whatever the name of the chromatography is, then the protein with that charge will stick to the stationary phase and elute last. So when it's an anion exchange chromatography, the anions will stick to the stationary phase, those beads, and they will elute last. So what charges the beads? If the anions are the last one to go, what is the charge of the beads? It's positive. So our stationary phase of this anion is going to be positively charged. So our positive charged proteins elute first, the anions get stuck behind, and they elute last. So that's the way. So if I think of cation exchange now, I'm not even looking at the slides, right? If I'm thinking of cation exchange, well, I know that the cations elute last. So that means that the beads are negatively charged. So right here, cation exchange means the cations will elute last. They will stick to the stationary phase. And in order for them to stick to the stationary phase, there has to be negatively charged beads. That's the way I memorize it. This is the way I don't get confused. Hopefully this helps between the distinction between the two. But that's basically essentially how you separate proteins based on charge, not size or anything like that. And then the last one is affinity chromatography. This is where you take more of the chemical properties, and it separates proteins based on specific binding interactions between the mobile, so like the solution that you're putting, and the stationary phase. So receptors on the stationary phase, so that's connected to the column, that match this target protein in the mobile phase is going to bind together, and it's going to basically be bound to the column. And then what ends up happening is you have all this like junk, these molecules that you don't care about, that's not of your interest, these proteins are not of interest, that are going to elute first, and you get rid of that. And then you're going to say, well, if it's bound to the column, how does it get unbound? How does it unbind? Well, we introduce a washing solution that will replace it. And I know it's not shown here, but in terms of elution, we're introducing like this washing buffer that's supposed to take its place of these like hexagon proteins, whatever. It'll take its place and it'll remove it, and then these then begin to elute. So in a way, based on the affinity and all that, it'll stay stuck like this. The reds will stay stuck to the stationary phase, but we have to introduce a washing buffer that will displace it, and then it begins to elute. So it's never the instance of like these will just naturally come off on their own. No, usually we have to introduce a washing buffer. That's usually how we do this fast. So here's a practice problem. The answers are in the footnotes, but this is something to do. Like, what are possible ways you can separate mixture of proteins 1 and 2? So here's some data about it. Like, does it bind to stationary phase? What's the size? What's the isoelectric point? And you just have to list ways that you can separate it. And then what would that look like in terms of the chromatography you run through it? So definitely go through these examples. It's very helpful. The answers are in the notes section. Now moving on to like Western blots, antibodies, all this complicated stuff. I'll give you an overview. So antibodies are proteins that bind to specific protein sites. So essentially, this is an antibody up here. And you don't need to know this like light chain, heavy chain, the hinge. You just need to know that there are antigen binding sites. They almost act like hands. And they're very specific to specific proteins, right? If this is the best analogy I can use. Let's say I have my hands together like this, right? If I were to try to catch, I don't know why it's showing the hearts. If I were to try to catch a football, I'd probably be successful, right? Like much more successful than trying to catch a baseball, right? A baseball would probably go through and hit me in the face just because I have it like that. But a football, because it's bigger, it'll basically like, I'll catch it. So it's in the similar way with these antigen binding sites. There's specific hands, specific conformations, and they'll bind to specific proteins, but not others. So that's the way you can think about it. So antibodies are this thing. So antigens are proteins that antibodies bind to. So the antibodies will bind onto proteins, and those proteins are synonymously called antigens. And in the body, antibodies are used to help remove foreign matter. So these antibodies right here are going to bind onto foreign material, foreign molecules, viruses, bacteria. They're going to form aggregates, and then the body handles it accordingly through your immune system. Okay, and so moving on to Western blots. So we're going to use fluorescent antibodies to identify specific proteins on a gel. So in this instance, we just done a Western blot. We have a bunch of proteins that are all found within one cell. Let's just say we mushed up one cell in solution, and this one solution now has 1,000 proteins that we don't even know how to separate, but we put it through this Western blot, and Western blots are used for separating proteins, and we separate them based on their sizes here. Well, how do we know which one is the right protein of interest that we'd like? Well, that's where we use our specific antigen system, this is where we use our antibodies to bind onto the protein of interest. And the antibodies, it's a two-step antibody system where we have antibody 1 bind onto the antigen, which this antigen is located on the protein of interest. And that's the first antibody that we introduce. It binds to it, and then we introduce a second antibody. But this one is going to bind only to the antibody number 1, not the antigen. And the second one, we usually incorporate it because of this GFP. It may sometimes alter with the function of the antibody, so we usually use 2 antibodies to do this method. But essentially the second one binds, and now because it's bound to that protein of interest, it'll only fluoresce whenever that protein of interest is there. So that's why we get this very specific fluorescence. And here's a real life example of it. We get an expression of this one, but none of the rest, because those aren't our proteins of interest. So that's how Western blots work with fluorescent antibodies. And then there's an ELISA enzyme-linked immunosorbent assay. And essentially it's an enzyme and antibody work together to produce a color change. And we have a target protein that's bound. So first we insert a capture antibody, or we incorporate one into the apparatus. We throw in our target proteins, we throw in a solution. The target proteins bind onto that capture antibody, and then from there we're going to introduce an enzyme label detection antibody. What this means is that it's an antibody with an enzyme function at one of the other ends that it's not supposed to bind to the protein, and then this is supposed to bind a just random reaction that when we introduce the reactants or the substrates, they're going to turn into products that are going to be colored. And so the color change indicates the presence of a protein of interest. So if we didn't have this protein right here, this antibody would not bind to the capture one. It would not. So then we wouldn't be able, because normally we would wash it, right? What washing means is we're introducing a water saline solution, and it's supposed to get rid of this antibody if it's not bound to the capture. Because again, the capture is embedded into the apparatus. The protein will basically favorably bind to this capture antibody. No matter how many times you try to wash it away, this target protein won't be washed. And then we introduce the enzyme labeled detection antibody, which if we then wash this away, or if we introduce a washing solution, this antibody will not be washed away at all because it binds favorably to the protein. And so even after we wash, the only things that will stay together is this complex. And then that's when we introduce this reaction for the color change. And then there's amino acid detection by UV visualization. I can't remember the full name. But recall from the amino acid lecture that phenylalanine, tyrosine, and tryptophan are aromatic. And with aromaticity, one of the properties is that they are also optically active, meaning that they can absorb wavelengths of light and be able to reflect out, or absorb the specific wavelength of light, and it wouldn't emit through a solution. So if we were shining light through a solution, like a little cuvette, some of the light would be absorbed, and it wouldn't be reflected onto the detector. And so the detector would say, oh, we're lacking absorption at 250 or whatever. Or sorry, we're lacking light at what's it called? At 280 for tryptophan, at around 275 for tyrosine, and then at 255 for phenylalanine. And then it would detect, again, this absorbance because it takes the inverse. Complicated stuff. All you got to know is that the higher the absorbance, the more the amino acid, that ring absorbs the light energy. And so, again, this feature of aromaticity, of light absorbance, we can use this feature to identify proteins containing these amino acids. And so typically it's 280 nanometers in the UV region for tryptophan, tyrosine. Phenylalanine, you can consider that, but it's more of a rare case, 250. I've seen on my exam, I believe, a while ago, predict what wavelength of light gets absorbed by tryptophan. And that's where you had to have like kind of this graph memorized where was the highest absorbance. You had to give like a approximate value. It doesn't be exact, but yeah. Something to keep in mind is that aromatics can absorb light. Tryptophan is the best at it. Tyrosine is the second best. Then phenylalanine is like the worst at absorption. And then we have the Lambert-Beer law of absorbance, and this one we use a spectrophotometer to measure absorbance of amino acids. We identify molecules and measure concentration. The only thing you really have to know is in this red box, A is equal to epsilon Cl. Epsilon is the molar absorption coefficient. C is the molar concentration. L is the optical path length. There's these other equations as well, like log is the initial intensity divided by the final intensity, like transmittance, all that stuff. And then there's also this one of absorbance is equal to 2 minus log 10 and percent T. Like there's all this extra equations, but realistically the only one you have to know is this top one. And know the units, know how they cancel out, and do some like practice problem examples. There's some online for sure. Maybe there may be one in the problem sets. I haven't looked too into it. So that's that. And then there's circular dichromism spectroscopy, which just means that it's a technique used to identify secondary structural characteristics of a protein. Are there helices, sheets, or randomness? And so that's what this is telling you right here, is that this red line is your alpha helices. So memorize kind of like the shape of it. That's all it's asking. Memorize the shape of a beta sheet as well, and then memorize this like black line, which is just randomness, random coil, or just a random secondary structure. And you may be presented with just one of these curves, right? And it'll be very distinct, and it'll tell you like what's the secondary structure of this protein that was put through CD, and then you have to say, oh, it's an alpha helix or something like that. It just provides general information about our entire peptide, but not super specific information about section to section of that peptide or within that polypeptide. And then there's peptide fragmentation. There's a class of like peptide proteases, which are just enzymes that cleave peptide bonds after specific residues. So for example, this will be covered more in depth in lecture 6, but trypsin is a protease and it cleaves after the arginine and lysine residues at the C terminus. This is just me. I don't know if it's at the C terminus or not, but it just cleaves it after or before. It can be either or. But again, we'll go into this in lecture 6. Just know that the peptide fragmentation is a thing that we use to isolate fragments and be able to detect it. And there's an honorable mention of Edmond degradation. This will be on your MCAT, so it's good to know, where it takes off one amino acid at a time from the N-terminus side. So you work from the N-terminus, take it off one by one by one by one, and then you'll be able to determine your full polypeptide based on that in the specific order. And then the final portion is determining protein structure. So we have X-ray crystallography, protein NMR, cryo-electron microscopy. All three of these, in their own respective ways, their own methods, will produce like a similar outcome, which is determining that protein structure. So X-ray crystallography, the way it works is it uses X-ray beams to generate diffraction patterns, which can calculate the electron densities. No, they're not going to give you a diffraction pattern. You have to do it yourself. It's just, you just have to know that it does this. And the pros is that there's high resolution and no size limit. However, you need crystals and no binding dynamics, meaning that the complex that you put into X-ray crystallography has to be stable. It cannot be this like transient, like transition state. We know that transition states, they don't preserve themselves. We can't just put it in a bottle. So that's something to know. Protein NMR, it maps resonance of nuclei in a strong magnetic field to gain structural information. So nuclei behave differently if they're dimagnetic, paramagnetic, all this stuff. But we know that if we put them in a strong magnetic field, they have their own characteristic shifts, which is what ppm means, parts per million. And these are just shifts in nitrogen. And the pros is that we see conformational changes from binding and in solution. So these nuclei act differently if they're bound to another molecule, or they act differently when they're alone, right? And some of the cons is that it's limited size and very extremely long runtimes because it's super sensitive. It's one of those things that it takes a while to run through and to get sufficient results too. And then the next thing is cryo-electron microscopy, where we use an electron beam to visualize large complexes or organelles, meaning that we basically almost like freeze the proteins and we make it extremely cold, because we know that, and this is kind of speaking to the second law of thermodynamics, where at absolute zero, the molecule is completely still. Crystal structure is perfectly still. So it's almost like that principle. So we freeze it so that the molecule stays still, and then we subject it to electron microscopy because it's even higher resolution than X-ray crystallography. So the pros is that we can do this with large complexes and visualize like channels. Right here, I think this is, what's it called? The pyruvate dehydrogenase complex. yeah, the pyruvate PDH. I'm pretty sure. Don't quote me on this stuff. And then the cons is that there's no binding dynamics though. So when we freeze it, again, that principle of it's static, we don't see those binding dynamics. Everything's like static. And when we say dynamics, it means like these transient changes of binding. What stuff gets bound to it and then released right away. So that's what we talk about that's what we mean by when we say binding dynamics. And then last but not least is like protein sequencing. We use mass spectra to determine the makeup of complex mixtures, determine the functional sites of proteins based on conserved amino acids. I mean, this is where we take stuff from wet lab and transform it into translational research. So it's to understand biological relationships, evolution across multiple organisms to inform now our current state, like at a Homo sapien level, essentially. And then here's a practice problem. How many peptide fragments will exist after protease A meets this chain? And then I've given you it cleaves on the carboxyl side after alanine and arginine. So you also need to determine from this peptide, where is the N terminus, where is the C terminus? You need to determine, okay, if it cleaves the carboxyl side, which direction is that? You need to find out what the amino acids are, where are the alpha carbons. And so if I were to give you a suggestion on how to do this to make it fast, just do like little circle the red with like a different color on your iPad, like the alpha carbons, circle them, and then from there determine what the amino acid is, and then determine N to C, and then from there, okay, I know it cleaves after alanine, so wherever it says alanine, cleave it. And so I'll play the answer here. You can pause the video, but this would be the answer. And this questions for me is just email me at Alex_Hernandez_Manriquez@brown.edu. It's on the first slide. Also, I'll be hosting office hours on Wednesdays. Still need to confirm the time and all that stuff, logistics, but I will be having it on Wednesdays. So yeah, just come up to me office hours. I know I talked really fast, but that's because I needed to cover a lot of lectures to get ready for also presenting lectures 4 and 5. But anyways, keep studying and know that this exam will probably be more applications based. I've told everyone in office hours that the first exam is usually like you learn the fundamentals and then you apply them to new situations. So just be ready for that. Exams 2, 3, and 4, those are perhaps a little bit more memorization heavy just because you need to know the pathways, all that stuff. But this exam, like, make sure you knock it out of the water, understand the fundamentals. If you're having trouble with it, come talk to me. Like, go talk to the TAs. Like, we're all here to help you succeed and knowing the fundamentals, because this will help you with your MCAT. This will help you with just in general upper level advanced biochem, chem classes, all that stuff. Take it from me. Like, I've had to learn this a couple times, and just knowing the strong fundamentals makes it a lot easier to relearn it when I needed to, and it just made studying for biochem a lot more enjoyable as well. So don't worry about the little teeny tiny details, like all the stuff I said about memorizing the residues per turn, memorizing like L amino acids are the only ones. Those are minuscule details. Like, at the end of the day, like, you can learn that pretty quickly in your dedicated study period. But as of right now, when you're more on like just trying to keep up, make sure you do the homeworks and all that, just learn the principles, and then the memorization stuff will come later. Like, just trust me, that's the best way to study and the most efficient. But anyways, I hope you all have a great rest of your week. I'm looking forward to seeing some of you in office hours. But yeah.